Hello my friends, how are you? Today I have very interesting topic especially for master and PhD student who just began in doing research and project in genetic engineering and molecular biology. The topic is unlocking genetic mysteries, discovering an intriguing gene. I hope this topic to be interesting for you and useful. Please follow me to the end of the video. If you have any comment, please write. Please share and subscribe and follow me for future videos. As we know, unraveling a candidate gene is a starting point of any project and research. There are a lot of excellent researches were published in very reputable journals, and they successfully figured out and fished out a causal gene. Indeed, there are many approaches that can be used for this purpose. All of us know that eukaryotic and prokaryotic genomes consist of a chain of nucleotides. Now I don't want to focus in the biochemical structure of nucleotides and DNA, but I would like to show this slide to show that the gene is a chain of nucleotides and the genome consists of coding and uncoding sequence. Decades ago, hundreds of plant genomes were sequenced. Sequencing plant genome raised huge data that deposited in several databases. We should note that sequencing results will show us billions of nucleotides: adenine, cytosine, guanine, and thymine. So our task is to figure out the coding and encoding sequences. Indeed, we need to decipher the raw data. How do we find a gene? We have to know that what are the genes, what do they encode, and how are the genes regulated. So there are several approaches that can be used basically for finding a gene. Firstly, by comparing the protein sequences. Also, we can compare with cDNA sequence or by genomic comparison. Gene finding refers to identifying a stretch of nucleotide sequences in genomic DNA that are biologically functional. Computational gene finding deals with algorithmically identifying protein coding sequence. So gene finding is not easy task as gene structure can be very complex. The coding sequence of a gene is a DNA sequence that can be translated to a protein. The coding sequence normally started from start codon ATG that can be translated to methionine residue and can be ended by one of a stop codons, either TAA, TGA, or TAG. DNA molecule can be transcribed to RNA, and some of RNA molecules can be translated to proteins, and these proteins can be function together, leading to a phenotype. Actually, every molecule can be sequenced in different approaches, leading finally to huge data for bioinformatics analysis. For example, a biotech trace will produce a signal. This signal can be perceived in the cell, leading to epigenetic response. This response can regulate the transcriptions. Many of RNA molecules can be translated to the proteins, and these proteins has many functions that can be interact together to regulate that metabolic productions, leading finally to tolerant phenotype. Actually, every event in this response can be sequenced by different approaches, leading to huge data that can be visualized by bioinformatics tools, finally to identify candidate genes and proteins for further studies. As I said before, we can find a gene by sequence comparisons, either DNA and chromatin. The concept of this approach is coming from the fact that the organisms share common ancestor, and the protein and DNA sequences are maintained and preserved during development and evolutions. DNA sequencing results showed that the plants and human being genomes are almost 50% are similar. Several studies showed that the protein sequences that are important for living organisms are conserved during the development. For example, the transcriptional evolutionary proteins are conserved in along organisms. So, such kind of important sequence are conserved during the evolution and development, and so we can find several proteins and DNA sequences that are similar in different organisms. The previous information led us to two concepts, the protein domain and the conserved domain. The protein domain are distinct functional and structural unit of a protein as shown here in the figure. There are here four types of domain, and the conserved domain are a unique domain in the protein structure that can be found in different proteins in the same organisms, and also can be found in different proteins that have the common function also in different organisms. So this figure is showing that the conserved domain can be exist in different type of proteins, and also the conserved domain can be shared in different other conserved domain to make a new proteins with a new function. So different kind of sequencing data can be deposited in web server, and by the aid of bioinformatics tool, this raw data can be visualized and we can understand this information and pick up our interesting genes. To work in database, firstly we have to understand the format of the database. Normally, the FASTA format is very important and very common used in different databases. The FASTA format consists of four components. A line of description. This description consists of a greater sign and a gene name, and also consists of DNA or a protein sequence. Now I will show important databases in plant science that use to find out a candidate gene. Phytosome is very important as of release version 13. Phytosome hosts 400 assembled and annotated genomes. Actually, it has several tools. We can determine the organisms that can be used for finding out our gene. We can find a gene by keyword, by the name of the gene, by its gene ID, or by a sequence. The National Center for Biotechnology Information, NCBI, is very important tool that has a gene bank, and it has a lot of database can be used to find out an interesting gene. In plant science, every plant species has its own database. Their database is a database specialized for Arabidopsis. Actually, this database is very important because it's the first database established for plant and has so many information. We can use this database as a reference to find out the function of many proteins and DNA that not annotated in other species. As my major is a citrus, so also in citrus there is an excellent database. This database is a citrus pangenome to breeding database. This database is based on pangenome. It includes 23 genomes of 17 citrus species, so we can compare the gene sequence and the protein sequence in different species, so we can figure out and decipher some important and interesting genes that not exist in other species, so we can pick up some genes related to our phenotype easily. So now I will show you how we can use the gene ID to find out an interesting gene. Also, I will show you how we use the DNA sequence and the protein sequence to find out our interesting gene, or maybe we can use a protein sequence from another database to find a interesting gene from another database. And now I will show you different types of database. So I will show you now TAIR database. This TAIR database I told you before is specific for Arabidopsis, and also I will show you citrus pangenome database. This is very important database who is working in citrus, and also here there is Phytosome database. This database I said there is here 400 genome. How we can use this information to find our interesting gene. So we'll try first by selecting tomato genome, Lyco, maybe you can like Solanium, Lyco, Persicum, tomato, this database, so we can retrieve some genes from some some papers. I will show you here. I retrieve some gene ID from papers, so I can copy this gene ID and go to the phytosome. So as here, first I selected the genome, and then we can find a gene by keyword. So we can use a gene ID to find our gene. So now we found this gene, domain of unknown function. We can go further for view gene report, so we can go further to find more information about this gene. As here you can see the gene ID and the transcript ID and the location in the chromosome, and also the description. This protein has domain of unknown function, but if we found more, if we search for more annotation using different database, we can see this domain. It has this protein has one domain actually is glycoside hydrolyse family 32. So this domain is unique and distinct for invertase proteins. Here you can see there is many databases used for functional annotation, and also here you can see some protein homologous in different species, and down you can see some sequences. If you want to use the genome sequence, you can use. If you want to use transcript sequence, also you can use. If you want the CDS sequence, also you can find here. And also here in CDS sequence you can see this gene consists of 1 2 3 4 5 6 exons. uh, and also this is a protein sequence. I like to use the protein sequence to retrieve some genes, so I will use the protein sequence of invertase gene from tomato to use. I will use this sequence to find out some homologous gene in citrus. Actually, let's do home. This citrus pangenome for breeding database. If you are working in citrus, or maybe you are working in different uh, species, almost most of database working in the same way. So we can search for some tools, or as I say, I showed in that database, you can find a gene by gene search by gene ID, or maybe by making blast. As I select protein sequence, so I have to select blast P for protein sequence, so I can use a protein as a query to find protein sequences. Also here I can find here this database, it has about 27 species genome, so I can select citrus protein 2 version 2, and I can blast the protein sequence, and this is what we say is a format. FASTA format, it has it consists of one, a first line is a description that consists of a greater sign and the gene ID, and then the protein sequence, and then we can click run. We will wait for a few minutes to find some hits. Here you can see, based on the protein sequence of invertase in tomato, we found this is a query sequence, and we found here many proteins that have the same conserved domain as showing here. To confirm really they are the same, have the same conserved domain and has the same function, we can go further to open the gene. Here open link, wait some time. So now we can see the location of this gene in the chromosome. We can see the transcript structure, it has different transcript, it has different exon and intron structure. Maybe we can retrieve some that promoter sequence, maybe some orthologous genes in the same species in citrus. Maybe you can see the gene expression, some key EG orthology, and maybe you can also study some function. Also, as I said, in Phytosome here there is the conserved domain based on many databases like Smart Gene 3D. However, I will show this database later on. But however, you can see that this protein also has the same conserved domain, glucosyl hydrolase domain 32. So this is showing that this protein I found here also belong to invertase family. Also, if I want to use another database like TAIR, this TAIR is a special database for Arabidopsis. As I said, it has the same background, so we can make search in blast. Here is a blast, so we can select blast P as I use amino acid query to search amino acid sequences. So here is a blast protein sequence in the FASTA format, and then we can run a blast. Wait a few minutes. Now here is showing some hits. You can see here the annotation of these hits. All of them belong to vacuum invertase 1. So maybe I can click one gene. I can show you what we can see inside. So this is annotation of that gene is vacuum invertase 1. There is actually in TAIR database there is more information and more functional annotation. There is this is the domain glycosyl hydrolase 32, and also here you can see the gene expression in different tissue types, and the gene location and biological process of this gene. And I hope this information is useful for you to find out your interesting gene. I hope you can follow me in Facebook and YouTube, and please share this channel to your friends, subscribe and write a comment if you have any comment for me, and you can contact me anytime by my email bashardahru@gmail.com. Thank you so much.